So I want you to just imagine for a second the sheer scale of the global tuberculosis crisis.
Oh, it's staggering.
Right. Like specifically multi-drug resistant TB. It caused, I think the number was 160,000 deaths worldwide in 2022 alone.
Yeah, that's exactly right. And it's an incredibly frustrating problem for chemists.
Because for decades, finding new antibiotics for this has basically been like looking for a needle in an endless, completely redundant haystack of chemical compounds.
Exactly. I mean, traditional high-throughput screening, which is what the pharma industry uses, it's just really sluggish. We test millions of compounds, but human chemists tend to look in the same familiar corners of chemistry.
Right, like tweaking the same molecules we already know.
Yeah, exactly. And meanwhile, the bacteria are actively mutating, outsmarting those familiar shapes, and building resistance.
Which perfectly sets up the source material for today's deep dive. We are looking at this fascinating 2025 research paper detailing something called Fleming.
Yes, Fleming. It's an artificial intelligence agent.
Right, but not just any AI. It's designed specifically to discover completely novel antibiotics for Mycobacterium tuberculosis, or MTB.
Which is the bacteria that causes TB.
Exactly. And our mission today is to explore how this AI isn't just, crunching numbers in the background. It's literally acting like a virtual team of scientists to design entirely new real-world drugs.
It's a massive leap forward.
Okay, let's unpack this, because if you are listening right now, even if you aren't a scientist, you need to know why this matters to you.
Right, because it really does.
It does. You are witnessing a real-time revolution in how life-saving medicines are discovered. This is democratizing a process that usually takes pharmaceutical giants billions of dollars.
Yes.
And decades to achieve.
Decades, yeah. And to understand why this is such a big deal, you have to look at why standard AI needed an upgrade to solve this.
Because we've had AI in chemistry for a bit, right?
We have, yeah. But up until now, those AI models were basically just really advanced calculators. You'd feed them a chemical structure and they'd spit out a single prediction like, yes, this kills the bacteria, or no, it doesn't. But Fleming is entirely different.
How so?
It's what we call an integrative AI agent. It's built on a large language model. Specifically, they used OpenAI's 𝗚𝗲 mini model for this.
Oh, so kind of like the ChatGPT people use every day.
Very similar underlying tech, yeah. But it operates using something called the REACT paradigm. That stands for reasoning and acting.
Reasoning and acting, so it doesn't just guess an answer and stop.
Exactly. It orchestrates this whole workflow. It actually manages 4 highly specialized subagents.
Wait, so it's an AI managing other AIs?
Yeah. You have the growth inhibition agent, the ADMET agent, the molecular optimization agent, and the generative agent.
I kind of like to think of it like a general contractor building a house.
Oh, that's a good analogy.
Right, so the LLM Fleming is the contractor. It doesn't necessarily do all the manual labor itself, but it knows when to call in the plumber, which would be like the toxicity model, or when to call the architect the generative model.
That is exactly how it works. And it has access to a massive toolkit. 9 different molecular AI models and 11 external tools.
11 tools? Like what?
Well, one of them is a literature search feature using something called PaperQA2.
Wait, it reads scientific papers?
It reads published scientific papers, yeah. So if it wants to know if a molecule is viable, it can cross-reference human literature.
That is wild.
And that agentic framework is a massive leap forward. Because it's not just predicting if a molecule works in a vacuum, it's balancing multiple, often conflicting biological requirements at once, just like a real human medicinal chemist would.
Which is crucial, because a drug that kills TB in a petri dish but melts your liver is
completely useless.
Right. So, okay, to be a good contractor or manager, you need a smart team. Let's talk about how the primary brain of this operation was trained, the inhibition model.
Oh, the training data set for this was enormous.
Yeah. The paper said they physically screened 114,933 diverse compounds and fragments.
Over 100,000, yeah.
And the hit rate for actually inhibiting MTB growth was only 5.7%.
Which just shows you how much of a haystack we are dealing with.
Seriously. So how did the AI process all that?
They use their top-performing model, which is called an EDMPNN.
EDMPNN. That is a mouthful.
Yeah, it stands for Evidential Directed Message Passing Neural Network.
Okay, I think we need to break that down.
Basically, the important part there is the evidential piece.
Yeah.
This framework allows the AI to capture uncertainty.
Uncertainty.
Yeah. It mathematically evaluates how confident it is in its own prediction. And the researchers set it up to filter out any low-confidence predictions, specifically anything with a confidence score under 0.6.
But wait, let me push back on this for a second.
Sure.
Isn't AI supposed to be, ultra confident? I mean, we use computers because they give us exact answers. Why is it a good thing that this AI essentially raises its hand and admits when it's unsure?
What's fascinating here is that in the noisy world of biology, knowing your blind spots is exactly what prevents you from wasting millions of dollars testing dead-end drugs.
Oh, because normally an AI would just guess anyway.
Exactly. A traditional model might look at a weird alien chemical structure, have no idea what it is, but still force a prediction. And if it's wrong...
You waste months in the lab trying to build it.
Right. But by capturing uncertainty, Fleming just says, I'm not sure. Let's skip this one. And it makes the predictions it's confident about incredibly accurate.
which we see in their perspective test.
Yes, the perspective test was amazing.
They gave the model 435 totally unseen molecules, right? Things it had never trained on.
Right. And out of those 435, the model confidently predicted that 6 would be inhibitors.
Just 6.
Just 6. And when they actually tested those in the physical lab, 5 out of those 6 actually worked.
That is an 83.3% hit rate.
Which is unheard of. A generic random screening hit rate is usually between 1 and 5%.
Wow. Okay, so it's incredibly good at spotting a cure if it already exists in the haystack.
Right.
But what if the haystack is empty? I mean, what if we need a totally new drug? That brings us to the generative side of Fleming, the chemical artists, basically.
Yeah, the generative models, because it can invent brand new molecules from scratch.
And it uses 2 different models for this, right? The first one is called 𝗬𝗏.
Right, 𝗬𝗏. It builds molecules using known chemical fragments.
So it's kind of like building a house with prefabricated Lego walls. You're taking safe, understood pieces of chemistry and just snapping them together in new ways.
That's a great way to look at it. It ensures the molecule is actually feasible to build in a lab.
But the second model is a bit more wild.
Oh, yeah, the conditional equivariant diffusion model.
Right. So if 𝗬𝗏 is Lego blocks, this diffusion model is like 3D printing a bizarre custom shape completely from scratch.
Exactly. It builds highly novel molecules in 3D, literally atom by atom.
Which is amazing, but, and correct me if I'm wrong, doesn't that require a ton of extra safety checks? Like, if you invent a totally alien chemical shape, how do you know it won't just collapse or, worse, hurt the patient?
You're absolutely right. And that brings us to ADMET.
Yeah, ADMET.
Right. It stands for absorption, distribution, metabolism, excretion, and toxicity.
Basically, all the ways a human body processes a drug.
Exactly. A drug that kills TB is completely useless if it destroys the patient's liver or if it can't even be absorbed in the gut.
So how does Fleming handle that?
Well, Fleming's optimization agent takes these generated designs and runs them through 37 distinct ADMET models.
37 different safety checks.
37 of them, just constantly testing and refining. And if we connect this to the bigger picture, this is where the AI really shines.
How so?
Because initially, the failure rate for these brand new AI-generated designs was around 42.5%.
Meaning almost half of them would have been toxic or useless in a human.
Right. But by running them through the optimization agent, tweaking the atoms to fix those ADMET issues, Fleming successfully dropped the failure rate down to just 5.0%.
Without ruining their ability to actually kill the TB.
Exactly. It maintained the antibacterial properties while stripping out the human toxicity. It is revolutionary to optimize for 37 human toxicity and metabolic factors before a physical chemical is ever even mixed in a beaker.
It's mind-blowing. But, okay, I have to play devil's advocate here.
Go for it.
Simulated success is cheap. I mean, you can make an AI say whatever you want on a computer screen. The real question is, did these AI hallucinations actually kill tuberculosis in a real physical lab?
That is the ultimate test. So the researchers set Fleming to run in batch mode.
Batch mode, so it was just running autonomously.
Yeah, completely autonomously. It generated thousands of molecules, ranked them, and then it used its LLMs to filter them for plausibility.
Like checking its own work against human knowledge.
Exactly. And when human medicinal chemists reviewed Fleming's final list, they actually agreed with the AI's logic 76% of the time.
That is a huge vote of confidence from human experts.
It really is. So they selected and physically synthesized 6 brand new, highly novel molecules from that list.
6 molecules that literally did not exist before this AI dreamed them up.
Never existed before.
Here's where it gets really interesting. They take these 6 brand new drugs into the lab and PLAB. All 6 of them successfully inhibited the MTB strain.
A 100% success rate?
Every single one. And it wasn't just a weak response, right? They inhibited it in a dose-responsive manner. The paper mentioned the EC50 values ranged from 3.6 micromolar to 75.4 micromolar.
Yes.
Which for our non-chemist listeners just means it was highly potent. It actually worked exactly like a drug should.
I really need to emphasize the statistical improbability of this.
Please do, because the math is crazy.
The chances of hitting 6 for 6 randomly on novel compounds is less than 1 in a million. Mathematically, it's between 10 to the negative 10th and 10 to the negative 6th.
Basically impossible without the AI.
Practically impossible, yeah.
Yeah.
And furthermore, 5 out of those 6 passed all of Lipinski's rules for drug likeness.
Which are like the physical rules for whether a molecule can actually be absorbed as a pill.
Exactly. And they also showed favorable pharmacokinetics, like robust human liver microsome stability.
Meaning the human liver wouldn't just chew the drug up before it could fight the TB in the lungs.
Right. They are incredibly viable drug candidates.
So we really need to take a step back and look at what we've just covered. Because we started with this massive, redundant chemical haystack that human chemists have been struggling with for decades.
Right.
And we introduced Fleming, this smart, uncertainty-aware manager, It's not just a calculator, it's an AI that orchestrates a whole team. It filtered out the bad guesses, it generated completely new shapes.
Optimize them against 37 safety models.
Right, and it literally invented 6 brand new, completely effective TB-fighting molecules, 100% success in the lab.
It's just a flawless execution of agentic AI in the real world.
And here is why this matters to you, the listener. Fleming is an open source tool.
Yeah, that's huge.
It has a natural language interface, meaning this totally lowers the barrier to entry. You don't need a massive pharmaceutical budget anymore.
You don't. Smaller, under-resourced labs can use this to hunt for cures for neglected global health challenges. They don't have to wait for giant corporations to step in.
It's democratizing life-saving science, which actually leaves me with a pretty mind-bending question to ponder as we wrap up.
Oh.
Yeah, think about it. We've just seen that an AI agent like Fleming can successfully act as a synthetic chemist, a toxicologist, and a literature reviewer all at once.
Right.
So what happens when these specialized AI agents start communicating autonomously with other AI agents working on entirely different diseases?
Wow.
Right. Like, will the future of human medicine be negotiated by algorithms chatting with each other in the cloud, sharing insights that humans couldn't possibly connect?
That is a wild thought. But honestly, the foundation for it is already here.
It really makes you wonder. Maybe the haystack isn't the problem anymore, because we aren't just looking for the needle. We've built an AI that can just invent the needle from scratch.
Beautifully said.
Thanks for joining us on this deep dive. We'll catch you next time.